this is a little bit of a different facility from everyone else because we have a unique focus. Our focus is making drugs, developing drugs, finding drugs, and getting those eventually to the patient. Now, how do we do that? We start with you. This means we start with your ideas, we start with your vision, we start with your concerns that you bring to us, disease that you care about, and then we take that forward and we wrap that into a discovery, into a journey that we take you on all the way into the clinic. So I'm going to step you through the individual bits and pieces here. But before we get there, let's talk a little bit about what we aim to do as a facility. What we do want to do here is we want to bring you top-tier infrastructure, expertise, access to small molecule functional genomics libraries. We want to transform scientific discoveries into medicines. And we do this for the patients in California and worldwide. The other thing that we really want to do here is we want to teach you. We want to teach you how to discover drugs. We want to equip you with the skills that you need to be successful out there in the pharma world, maybe building your own enterprise, building your own company. This is where it all starts if you want to be within drug discovery, biomedical research enterprise. This is what we like to do. Okay, how do we do that? We focus on high throughput screening. It's a productivity tool and has many applications, but what is it? It's really the execution of the same assay at a scale that's industrial, more than 10,000 times per day. Now, let that sink in. More than 10,000 times per day, with reliability, with basically no fails. So, what can you do with that? Drug discovery. So we talked about that, we belabored that. We can screen all sorts of libraries, targeted drug-like diverse libraries, but there's a lot of really interesting applications that are maybe a little bit more up your alley if you're a basic researcher. One of them is functional genomics. You can use CRISPR, cDNA, SI, all of the tools that you're used to. You can do this in high throughput. Or chemical genomics, where you're not modulating the genome, but you are actually changing, blocking key pathways, signal transduction, etc., using small molecules. It's a very fast, maybe a little quick and dirty approach to discovery, but it's uniquely powerful. And then, of course, there's materials discovery, characterization, toxicology, etc. But today, I just want to focus on drug discovery, genomics, and so on and so forth. So, how do we give you access to high throughput screening? We give you access to a very unique environment that's roboticized. And you see this here in the middle, you see a robotic arm that travels up and down. Well, I wonder if we have something like a laser pointer. We do. So that travels up and down a little rail here and connects all of these things that we call peripherals. This can be incubators, this can be automated microscopes, plate readers, washers, dispensers, liquid handlers, you name it. We're covering everything that you might do by hand normally in your lab with automation. What this enables is, and what this gives access to is the ability to run your experiments 24/7, 365 without anyone having to be in the lab. Your productivity, and I'll show that in a moment, is super enhanced. Okay. The other thing that this gives you access to is really the ability to screen our small molecule libraries. We have about 330,000 very well curated small molecules. And just as a comparison, I think Gilead has about 750,000. They're getting slightly nervous because we're approaching their size. There's a little bit of overlap, so we are up there mid-size pharma level. It's very unusual to have some resources like these. But what really sets us apart from everyone else is functional genomics. Genome-wide, SI, now here's a lot of stuff that's cut off. There's CRISPR missing, there's cDNA missing. There's a lot of other things that I'd love to tell you more about if you come and visit me on the 6th floor. Just to show you give you a graphical impression of what this means to go from manual to high throughput. Just brought you what one user did with us. They were screening 10 IC50s dose response experiments in triplicate a week, and they went from that by hand per person to 250 in an afternoon. That's the difference. That's the difference from manual, automated. That's tens of thousands of data points in an afternoon without you even having to stand there. So, at the end, what comes out of that? What comes out of that are molecules like 3515, which is the first small molecule that is UCLA homegrown, that made it into phase 1 clinical trials for oncology. It's gotten finally orphan drug status. It's now moving into phase 1. I have heard that it now passed phase 1 with flying colors, which is exciting for us. But this is really what comes out of our research when it's successful. Another success story is DNP19, which is an antibody that you can load up with PET isotopes or with drugs or. It binds to LRC15, and that's a TGF-beta-driven biomarker. And you find this on the surface of very aggressive cancers. You see how this particular antibody here, you see this in yellow, gets internalized when you have a cell and when it binds to it. That's really important for such a drug because that means that this gets taken up in the tumor and doesn't come out anymore. What this also means is that it actually is able to knock down the tumor growth. You see this here in a mouse model of an osteosarcoma, where after one treatment at 30 megabecquerel, basically the tumor is completely ablated. It doesn't come back. 160 days later, it's still gone, which is an excellent result. Also, of course, importantly, you don't find this drug anywhere else in the body. So this is, again, a mouse model here. You see osteosarcomas, here HU09, SAO2, U118, different types of tumors. It gets taken up really, really well. But this is what it looks like when research in my book is successful. Okay, so I'm going to stop here and see if you have any questions. If you would like to get in touch with me, my email address is down here. And yeah, I'm looking forward to seeing you upstairs on the 6th floor. Thank you.